Okay. So to start us off, I just wanted to talk a little bit about I am ALS, and give you some context, and some of you may not be as familiar with us as others. I am ALS was founded in 2019 after Brian Wallach was diagnosed with ALS at the age of 37. Him and his wife, Sandra, really wanted to focus on building an organization that put patients at the center, and was really focused on engaging with policymakers and offering vital services for people impacted by ALS. So the goals that they sort of grew this organization around was to continue bringing people like yourselves into the movement, and continue to raise awareness about ALS and also competency in different demographics, whether it's medical professionals or the general public, improve the ALS therapy pipeline, and improve the quality of life for people living with ALS and their loved ones. So this was sort of the basis for I Am ALS. And so we've been working through our community teams, like the clinical trials team that is putting on this webinar, to focus all of our work on these four goals, sort of like our pillars. And so the clinical trials team is a team that that is focused on supporting that therapy pipeline that is in our goals. And I am really excited to be able to continue talking about the clinical trials work, and want to make sure too that you all know that this clinical trials webinar is part of a series. So this is the fourth webinar that is included in this series, and we have another one coming up later this year that's going to focus on navigating clinical trial finders. But if you would like to find any of these other webinars that we have done, they are all on our YouTube channel, and I'll put the link to that in the chat. But before we dive in all the way, I just wanted to put some ground rules, some housekeeping items. If you could please mute yourselves if you're not already muted, to make sure that we can hear our speakers. Please feel free to put questions in the chat. I will be monitoring the chat. Our hope is to have time for question and answers at the end, but please feel free to put things in the chat as they come to you. If you have any specific questions that you would like that we don't get to, please, we will try and do our best to answer them in a follow-up email. So please, if we don't get time to actually do a question and answer, still ask your questions. And on that note, I am going to turn it over to Dan McIntyre.
Hi, thanks, Julie. And thanks for coming, everybody. It is a pleasure and an honor, and I really mean that, to be able to introduce Dr. Lyle Ostrow to this group. Dr. Ostrow is an ALS neurologist and researcher at the Lewis Katz School of Medicine and the MDA ALS Center of Hope at Temple University, which is a truly extraordinary organization. He's not only a scientist, but he also is devoted to the well-being of people living with ALS and their families. He's on the scientific advisory board for Everything ALS, and he's on the board of directors of the ALS Hope Foundation, which you probably know also is based at Temple. Dr. Ostrow is director of Temple University's ALS Postmortem Core, which is funded by a collaboration with the CDC National ALS Registry. And that's something that he founded at Johns Hopkins more than a decade ago. The core does one of the most important things that can be done relating to clinical trials. It provides de-identified clinical, pathological, and genomic data that are made broadly available to academic and industry ALS researchers, which is critical. He's also chair of the programmatic panel for the Department of Defense ALS Research Program, which is the largest dedicated annual funder of ALS therapeutic discovery and validation. He led the efforts to define and develop new mechanisms to help move novel preclinical therapeutics through the drug development process to emphasize biomarker development and de-risk and improve the design of clinical trials. And also really important to encourage open data and resources sharing. So that's the thumbnail. It doesn't do it. It doesn't begin to do service to what Dr. Ostrow does, but I'll let him take it from here. Thanks very much.
Wow, thank you, and thank you guys so much for inviting me today. Let me hold on. Let me make sure I can share my screen here first, and then
Just like we rehearsed it, right?
Hmm?
Just like we rehearsed
Yeah.
it.
You know, it's funny because of that, the postmortem core that you mentioned. I've actually been on Zoom for like the past 12 years because it's like that's the way that we communicate with other researchers using our resources. Can you guys see my screen now? Did that work?
Yes.
Okay. So thank you so much. I'm really, really happy to be here and talking to you guys today. And I hope I'm going to do what you're hoping I'm going to do, which is talk a bit about, you know, how we can all be responsible consumers of information about ALS clinical trials, about things that we read, press releases, and not just be consumers, but I mean, this is I Am ALS, man. So part of this talk is going to be how, number one, you guys have already impacted all this, but really what we can do together to improve the way that clinical trial results are communicated, to improve the design, and really educate our community. And, you know, I'll start, you know, we live in an interconnected world, right? Like we find out about stuff that's going on across the world almost instantaneously. And, you know, that can be anything from a sporting event to something political. And I do think that as a community, though, we are not harnessing this interconnected world we live in as well as we should, to accelerate the development of these ALS therapies. And maybe we'll talk about that a bit too. You know, so as you mentioned, I'm an ALS clinician. I take care of people living with ALS in my clinic. I have a research program. I have funding support from the NIH, a research grant for a particular therapy, and also from the CDC National ALS Biorepository, which funds our postmortem course. Sorry about that. And we're going to talk a fair amount today about some of these efforts, in particular the programmatic panel of the DoD ALS Research Program. And I already saw, I can't see the screen anymore, but at least a couple people who've just logged on who I know have been consumer reviewers for that program. So thank you. And we're going to talk about that. You know, that I want to be very clear. I don't speak for the CDC or the Department of Defense or any of these other efforts. It's a privilege and honor to get to work with them. So this is a slide I stole from Dr. Scheffner, who I'm guessing some of you know. This was from one of the clinical research learning institutes that Niels puts on. He says, what makes a successful trial? A drug with good preclinical science, adequate pharmacokinetic information. So what's that? That's, you know, what happens to the drug's concentration in the body and how long does it stick around? The appropriate dose, an easily measured outcome relevant to disease, an effective design, sample size and power that depend on the trial goals. That all sounds pretty straightforward, right? But what does any of that really mean? I mean, what is good preclinical science? Who decides, right? How do we know what the appropriate dose is? With ALS, one of our biggest challenges is what's the outcome measure? You know, if I am doing a clinical trial for a cancer, for a type of tumor, and I give somebody a medicine, I can see, did the tumor get smaller? Did it get bigger? Did it stay the same size? If I give someone a blood pressure medicine in a trial of, you know, high blood pressure, the blood pressure goes down or it doesn't. ALS, as you guys know, is a challenging disease to measure. And it's also a very protein disease. It's very different in different people. And so what is an effective design? And what does it mean, you know, sample size and power that depends on the trial goals? And I'll say this is an intro slide. Dr. Scheffner explains a lot of this as part of the Clinical Research Learning Institute. This is Nadia Sethi, who I know some of you also know, who also is faculty for some of the Clinical Research Learning Institute. She also serves on the programmatic panel with me for the Department of Defense and does 47 other things that, like many of you ALS warriors, I don't know how you do it all. This was a post on social media, right? Drug development is difficult. Drug development for ALS is even more difficult. What doesn't mean we won't succeed. Does mean we need trials now, patient-centric trials, but there should be a damn good reason for a drug to be in a trial, and strong preclinical science is crucial. So I mentioned these, I strongly would encourage anyone who hasn't participated in one of these clinical research learning institutes to do so. So there's a link at the bottom of the screen there. These were started by Dr. Bedlack, who I think gave the last talk in this series for you guys about the clinical trials. Certainly is one of my personal heroes, along with Dr. Hyman Patterson, who's right here. And, this is a 2-day course. It's free. You have to apply for it. And it is designed to teach you everything about clinical research, ALS science, clinical trials, how to be an advocate, how to help others understand these things in your communities. There's virtual ones, there's in-person ones. This particular one here, this was an in-person one in Philadelphia that, even though I'm in Philadelphia, I was virtual because I happened to be in New Mexico at the time. But check it out. They're really, really an amazing experience. And as an example, this is an agenda from that one that I just showed in Philadelphia. And I'm not showing this to say, here's all the stuff you have to learn. But I just want to point out a couple of things. And so one of them is that the quote unquote faculty is clinicians, it's scientists, it's people who are advocates, it's people living with the disease, it's people whose family members have had the disease. And I'm not going to teach you all this stuff in the next 30 minutes, right? This is a lot of info, but this is the kind of thing that matters to be a responsible consumer, to understand, how do we decide what we target, which we all give different talks on these. I didn't remember I gave that one. How do we design trials? What are important for statistics? How to read a research paper. There's an exercise on how to read a research paper. Dr. Hyman Patterson does these as part of the Curleys. I give a talk at this often on how to read a grant application, because we have people living with disease and their family members as reviewers for the Department of Defense. What's that experience like? And so again, it's something I would strongly recommend anyone who hasn't done it, that they think about it. And so Terry, Dr. Hyman Patterson, this is one of the talks that she often gives at these, right? And I put a link, there's a YouTube, some of these are recorded, some are virtual. I'm sure there might be a more recent one. I don't know, but this is the one I know about because it's on our Hope Foundation website. But she gives a talk about how to read a research paper. And her main point of that talk is you don't have to be a scientist to think critically, right? Anybody can think critically. Anyone can read a paper, and you don't necessarily have to understand the nuances of the biology if you ask the right questions. And so if you're curious, again, I don't have time to do a 2-day curriculum, but this is one of the talks that she gives. And then after that talk, as part of that course, there's a homework assignment. And this is a video too of that next session, where everybody's given a paper. And if you email me, I'm happy to send you this paper. And there's a questionnaire or critique. And it's basically, you have to read the paper and think about it. Think about, how is it designed? How many people are enrolled? How long was the trial? Is there a placebo group? Was it blinded? Did the investigators know who got the drug? What did they measure? What was the safety? How did they know if it was efficacious? These are the sorts of things that you can ask as you read these sorts of papers. And if you're interested, this YouTube video will take you through this one. So, Terry gives this talk at the Curleys, and her point again is that you don't have to be a scientist to think critically. I often follow that talk with a how to read a research proposal. As I just mentioned, this is my quote. I learned more useful skills as a beer salesman than I did in medical school. And so what do I mean by that? And so I'm actually like a certified beer judge, right? This is crazy, I know. But so my family owned a gourmet store like in Buffalo, New York, called Premier Gourmet. And I ran the beer department. It was my indentured servitude. And it probably added like 3 years onto my PhD because I was busy selling beer. But as part of that experience, running the beer department, I became a home brewer, and I befriended brewers and professional brewers. And I ran a beer conference for charity for the Roswell Park, beer conference, a beer festival. Wow, that's kind of funny. And the exam, I took an exam to become a beer judge. It was harder than any exam I had in medical school. And everything I knew about beer was taught to me by my friends who were these amazing professional brewers who knew a million times more than I did. But guess what? I was a medical student at the time, and I could write an essay and take an exam, and I could cram tons of information. And so I scored really well on that silly test, and I was embarrassed about it. And when I started judging beer competitions, I'm like, I'm not qualified to do this. Like, what am I? They don't care about my opinion. I'm not a professional brewer. And what I learned, and the thing that's very similar is, again, anybody can think critically. And the key is, when reading a grant, know your marching orders, understand the mechanisms, understand the criteria, and you get good at it and you get fast at it. And, just like this was last month, by the way, in Pennsylvania, I still judge beer. And it's really not that different of an experience. So when we evaluate research proposals that are preclinical science, these are the kinds of things we think about, right? And they're not that different than the questions we ask in a clinical trial, because in many ways, the preclinical work is supposed to be approximating a clinical trial. So what's the question being asked? Is there something that goes up or down in ALS or is different in a particular type of ALS? And how do we measure that thing? And, is there a clinical characteristic that relates to whatever it is that we're seeing under a microscope? And what happens next? So if the experiment's successful, what does that mean? And what's the outcome? And why does this matter? And is the experiment that's proposed specific to maybe a particular flavor of ALS, like a particular gene mutation, or people with certain types of biological abnormalities? Is the model or the samples being used appropriate? I mean, these are the sorts of things that we think about when we're looking at a clinical trial. Is there good biological rationale? Is there some reason for this treatment? What we call a target, right? Where do all these targets come from? Who decides? And we're going to get to that in a second. And is the model that it's being tested in appropriate to test that? Is there reason to think that what's happening in this laboratory experiment might be happening in people living with ALS? This is one that comes across a lot in both preclinical basic science, but unfortunately also in clinical trials, right? So oftentimes we talk about a drug in search of a disease or a technique in search of an experiment. What I mean by that is there might be a company that owns a particular drug, the patent on a drug that targets some mechanism. And so that's their intellectual property. And then what they do is they say, hey, let's see if we can find diseases for which our drug might work. And as you guys know, there's lots of different biologies that have been implicated in ALS. And so, you do a Google search. I probably shouldn't tell you this, but a few weeks ago, we had a drinking game, some friends, and here was the game, right? It was type in the word into Google Scholar, which is like PubMed, the word amyotrophic, right? So for amyotrophic lateral sclerosis, and then any cell type, chemical, cellular receptor, biological pathway that exists in the nervous system, like anyone, like anything that's on any cell in the nervous system. And if a paper came up talking about that in ALS, you had to drink. And let me just tell you, the game didn't last that long. It's hard. And so you have to be careful, is there good rationale in ALS? What's the rationale? Is there a good reason to do it? So, stepping back, how do we do this, right? These are the how does something become a target? And what does it mean? We always talk about, we have to validate this, right? And why are some things in trials and not other things? And I can't tell you the number of emails I get. And by the way, my email was on that first page. I'm happy to talk to anybody about anything, anytime, truly, sincerely. If I can help with anything, I work for you. It's my job. I work for you, and I mean it. And people on this Zoom who know me, I'm serious. Reach out anytime about anything. But this is the thing, right? People reach out to me and they're like, why isn't nobody testing this? And this sounds so exciting. And why isn't this in trials? And who decides? Who's the they? I mean, where does this come from? And why have so many trials failed? And one thing we hear a lot is, why can't we develop drugs like they do in cancer and HIV? How can we de-risk industry investments, right? All these trials that have failed, if I own a venture capital firm or even a big drug company, why would I want to spend money on an ALS trial? I mean, if it's going to fail. And so how do we de-risk these investments? How do we make it so that the industry will be more interested? And by the way, they are, and more so now than ever before. And why is that? We'll get there in a sec, I hope. And, we hear a lot about biomarkers. And so what does that really mean? The different kinds, how are they used in trials? Why do they matter? And so, the rub is this, right? And we all know this, that ALS is a clinical diagnosis, right? Again, there's no blood test aside from the people who have a genetic form of the disease that says this is your ALS level, and I'm going to give you a drug, and and this is how we diagnosed you. It's a clinical diagnosis. It is a diagnosis of progressive painless weakness, and it looks very different in different people living with ALS. It can go fast, it can go slow, it can cause what we call upper motor neuron symptoms or lower motor neuron symptoms. There's lots of different genes that can cause it. It can look different even in two family members. And so with all of that, it's hard to define what's a successful trial, right? And how do you know if a drug is working? And so, over the past couple of decades, we've learned an unbelievable amount about the biology of ALS. And yet what we've learned has largely failed to translate until very recently. We're now getting some signals, and it's really exciting. But we talk a lot about, are the models we're using in lab, is a mouse with a genetic form of ALS a good model maybe for somebody who doesn't have that genetic form of ALS, right? Or our cells that we're using in a lab, like nerve cells or other types of brain cells that we've gotten from people living with the disease, are they the right thing to be testing drugs on? How do we know if we're using the right treatments in the right people at the right times? And this is back to the de-risking thing. And I'll tell you, one of the things we did for the Department of Defense research program, I went to people in VC that we know, and we said, hey, what do small pharma and ALS screw up? What are they bad at? Like when they come to you and they say, can we, will you invest in our treatment? What are they not doing well? And the answer is always the same. It's what are called predictive biomarkers. So something that says that a particular person living with this disease is likely to respond or not respond to a given treatment, right? And so that's why I think there's been so much movement in the genetic forms of the disease, because on the industry side, they consider those forms of the disease to be de-risked, right? The predictive biomarker, if I'm doing a trial of a type of therapy that's supposed to fix a genetic mutation, the predictive biomarker is the gene test, right? It's, do you have that mutation? Qalsody, tofersen, the drug for SOD1, which, we're all very excited because it seems to be making a difference in people with that type of ALS, even more so than we thought from the trial. If we had enrolled everybody with ALS in that trial, not just people who had that mutation, the trial would have failed. And yet, if you think about it, that's what we keep doing over and over for sporadic ALS. And and maybe we know that that's likely the issue, because we know that there's different biologies in different people living with the disease and different biologies at different stages of the disease. And so if a drug is targeting a particular pathway, we need to do a better job of figuring out which people living with the disease should get which therapies. Otherwise, it would be like giving, a gene therapy to everyone who has the disease. And of course, we have to improve our measures to tell disease progression and different kinds of disease. Now, this is the thing that really gets, by the way, I know, I think when Dr. Bedlock gave you this talk, they talked about different drugs that are in development right now, and there's tons of them, and we all talk about them, and we all talk about which ones excite us the most and which ones we think are the most promising, right? Let me be very frank with you. Supposedly I'm an ALS expert, right? But what does that really mean? Because I understand, I can explain the biology, I understand the clinical picture, I can tell people, to maintain their weight and not fall and do all the things that we know are helpful in this disease. But I'm not going to tell you which one I'm most excited about, because if I did, I'm full of baloney, right? And we all are. Otherwise, over the past 30 years, we would have done a better job of choosing the right ones. What I will tell you is which trials are well designed, which drugs have good biological rationale, and what that rationale is, what the risks are, what the evidence is that might make this a trial, and how we're going to know if the trial is working. And then, my job is to educate you, and then you decide if it's the right trial for you. And this is the thing that is frustrating that we need to improve. So when a trial fails in ALS, we legitimately often don't know why. And the first example that really sticks in my mind when I was, kind of coming up in this field was ceftriaxone. And so this was, early 2000s, right? And so this was based on what was called the excitotoxicity, theory of ALS, that there's too much of something called glutamate, which excites nerves, and too much of it excites them too much. I'm not going to go through all the biology right now, but it was developed in a laboratory model, and then there was a couple of different ways that they screened all these approved drugs to figure out if any of them increased this thing called a glutamate transporter, which presumably would fix this problem. And they tested them in mice that had a particular ALS genetic mutation, this SOD mutation. This is a figure from that paper. This is the kind of data we often see in preclinical studies. So this is the percent of all the mice that are surviving and how long they're surviving. And so this is the placebo, saline, and this is the drug. And every time a mouse percent dies, this line goes down. And so what this is showing is that mice that got the drug, on average, lived longer than the ones that didn't. And then there were 2 phase 2 trials that looked really promising. There was a lot of excitement. And then it came to phase 3 and it failed miserably, so miserably that it was stopped early, right? And when it was stopped early and when it failed, we didn't know why it failed. Like, did it fail because it was just a dumb idea? Or maybe, it had been tested in this one kind of mice. Maybe that didn't generalize. Maybe the drug didn't get where it had to go and have the biological effect because it didn't cross the blood-brain barrier. Maybe it got there, but it just wasn't sufficient in increasing those transporters. Maybe it works, but only for some people, and we didn't know how to identify them, or at some stages of the disease. And the fact is, we still don't know. And we don't know why that drug failed. And the reason we don't know is that we didn't have the right kind of biomarkers to tell us who should get it, and did it have the biological effect it was supposed to have? Because if we'd had those things, we would then be able to know at least, was it a good idea or not? So around that same time, so this is Jack Griffin. Jack was the chair of neurology when I came to Hopkins as a resident. And he was my personal mentor. I was his clinic resident for 4 years. And he also was the editor of Nature Clinical Practice at the time. And he wrote a bunch of these, very short, really meaningful editorials. And this one, right around the time of that ceftriaxone trial, research in neurologic diseases lost in translation. Brain sciences are on a roll, young scientists choosing this for their careers. We're learning all sorts of stuff. Nevertheless, there is a palpable restlessness among disease advocacy groups, philanthropists, neurological investigators, impatience and frustration, effective treatments, right? Not for the most prevalent diseases are not emerging at the rate we would have predicted. And this was in 2006, right? Does it sound familiar? Can people here relate to this? I think we can, right? And it's kind of frustrating. And this short essay that he wrote had these conclusions, and they were mostly related to new ways to collaborate and exchange data and ideas and more rapidly fund research. And I'll tell you what, this is happening, and this has really been a dramatic change over the past decade and really moved things forward. But this isn't the entire answer. So fast forward, this is a review from just about almost 2 years ago, right? And so now, instead of lost in translation, it says poised for successful therapeutic translation. So, what changed? And this particular review makes some good points. One of them being that we've gotten a much better idea about the different genetics, the different mechanisms. We've got ways to identify when people are becoming symptomatic, what's called phenotypic conversion. We've got biomarkers that tell us if a particular drug has the biological effect it's supposed to have. Different ways to measure the disease, different ways to interact with these biologies, different genetic therapies that were science fiction even 10 years ago. And all of this is reasons to be excited, and reasons why the quote unquote pipeline is so filled with so many drug development programs in ALS right now. Now, I'll tell you though, all of these things are true. And also, by the way, new trial designs, and a lot of this is due to people living with the disease having a role in helping design these trials and making them more meaningful. This paper missed one important point, which I will get to in a second, which I think in my mind is the most biggest change. So how do we look for these therapeutic targets? Where do they come from? It's pretty straightforward, right? So in a lab, you have cells, or you have, an animal model, you have something that has disease and something that doesn't have disease, and you look and see how they're different, okay? And sometimes it's obvious, like sometimes a diseased cell or a diseased animal looks very different, like an apple and an orange. You can easily tell them apart. Sometimes it's not so obvious, like maybe two different, species of apples. And you can't tell them apart easily, but if you grind them up and if you make applesauce, and if you then analyze that applesauce, you're going to find, different biochemical components. And literally, this is how research is done, right? We grow cells, we grind them up, we analyze them, we look for changes in different proteins, different pathways, and then we say what's different about disease versus not disease. Now, one of the problems that we have with that, though, so let's say that I have an orchard, right? And some of my trees my apple trees are dying, the apples are dying, and I want to figure out what's different. Well, if I go and analyze the apples that are left on the sick tree rather than the ones that have fallen to the ground, maybe they have some protective feature. Maybe they're not the problem. If they're still on the tree, if I'm taking cells from an animal model or a person living with disease or looking at their brain under a microscope late in the disease course, maybe the cells that are left are actually ones that are protected. And these are the kinds of considerations we have when we do these experiments. You hear a lot about things that we can measure in blood or spinal fluid, right? Oh, by the way, the brain's not applesauce, right? If apples are nerve cells, the brain's not applesauce, the brain's like apple pie because there's lots of different kinds of cells in the brain. And as the disease progresses, the ratio of those different cells, different kinds of cells changes because you lose neurons and maybe you gain other inflammatory cells or other cells. And so if you grind up that apple pie and analyze it, you're going to see changes just because of the cell type changes. And if you measure something that's in the spinal fluid or the blood, that's not dissimilar to like this ice cream melting on this pie into the dish and picking up some stuff along the way. And does that matter? And so it's really important that rigorous science is done to figure that out and to figure out if something is meaningful. And, so you mentioned at the beginning, I run this postmortem tissue core, right? So when people, often my own patients, but people from around the world, pass away from ALS and decide to donate their bodies to research, it's my job to make sure that they are used responsibly and in a meaningful way and for as much research as possible, and that the data is shared broadly, right? And so that's this postmortem core. And the point is, if we see something in a mouse or we see something in cells in the lab, we need to see, is it also happening in the samples that come from people living with this disease? So, this is really the ultimate donation. It's the kind of thing that we would not be able to do this work without, and it really, really, really matters. So, and by the way, oh, I somehow forgot a slide. Was it not there? Oh, I'll hate it if it wasn't there. Ugh. I think it is. It fell out of my slides. I'm so sorry. So I had a slide that disappeared. That was a video of these mice running around. And the point of that slide, man, I don't know where it went. The point of that slide was that this was a particular drug that was being tried on. These are SOD1 mice. And maybe I can share the video with you later by email. But some of these mice were all litter mates. They were all born from the same mom, and they all got, they all have this SOD1 mutation, the same genetic mutation. And some of the mice were given placebo, some of them were given a drug. And if you've been able to see the video, what you would see is that there's mice who got the drug that are walking around a lot better than these mice. But there's also one of these 4 mice, he's walking around pretty damn well. And a couple of these mice, not so much. And you notice the mice look different, right? Some are dark, some are light. And the point of it is that, this is even in this genetically identical mouse population, these were all litter mates. Not all the mice are the same, and they don't all respond to the drug the same. And this is, just one kind of ALS. And my role in this project, I didn't do anything with these mice, but these were tissues from people who had died from ALS that we were providing and helping to analyze. And so the diaphragm is the muscle that moves air in and out of your lungs, right? Wild type is normal. This is the genetic ALS mice. And, see this yucky spots? That's the abnormality, right, in these mice with ALS. And what we did was provide human tissues, also from the diaphragm, from people who didn't have ALS, people who had sporadic ALS, people who had C9 ALS and other genetic forms, and we're able to show that this particular pathway, in fact, looked the same in people as it did in the mice, and looked the same in different kinds of ALS. So this is the sort of important information to help us figure out whether something might be a good therapy. Now, you guys know, as the years have progressed, we have implicated more and more and more and more genes in this disease, right? And the genes have all different kinds of functions. And the first gene, SOD1, which we finally have a treatment for, was developed back here in 1994, was when that was first published. And as our techniques have gotten better, we keep finding more genes. This, by the way, is a little puzzling, right? How can so many genes cause this specific disease? But as we've learned about all these different kinds of genetic ALS, it gives us different things to perhaps target. And this, by the way, this is supposed to be me. This makes my head explode. These are just some of the mechanisms that have been implicated in ALS, implicated by comparing cells or mice or different types of assays in a lab with and without disease, right? These are all very general cell mechanisms. Like we all learned in high school, mitochondria are the powerhouse of the cell, right? Like every cell in my body uses them. So how the heck can mitochondrial dysfunction cause ALS? But it does. And it's even worse than that, because all these mechanisms, we've learned there's different cells, like the abnormality doesn't have to be in the motor neuron to get a motor neuron disease. And most importantly, what we've found most recently is that there's clusters of people living with this disease, with sporadic ALS, in whom different mechanisms dominate. And so if you're somebody who has, say, abnormal microglial activation, and I give you a drug that targets ER stress, it's not going to work, right? Or it shouldn't work. And that's where we get to this idea of these predictive biomarkers. Oh, the slides were just out of order. Here you go. So these guys are all SOD1 mice. These got saline, these got the drug. Clearly, I think clearly, most of these guys are moving around better than these ones. These ones are pretty paralyzed. But there's at least one dude here who's not moving so well. And there was at least one guy here who's moving better. And so it just like in a experiment in people, which we call a clinical trial, you have to have statistics, you have to have the right numbers of mice, you have to be able to interpret, whether or not it was successful. Keep in mind, this is a very specific genetic form of the disease. If you expanded this to all people living with ALS, there would probably be even more variability. So as we've learned over the past several years, this is back to that article about the advances we've made. All these different mechanisms have been implicated in the lab. And look at, this is even out of date now, but these are all trials of different drugs targeting these different mechanisms. I mean, it's unbelievable how many different drugs are being developed for specific biologies. And these are all genes that have been found to affect these different biologies, right? And, again, it's the part we still don't understand is how all these diverse mechanisms and all these diverse genes can cause this very specific disease. We truly don't understand that. But what we do know is that there's lots of different flavors of ALS, and we need to do a better job of giving the right drugs to the right people. And in fact, this is 2023. This was an editorial right after the recent Relivrio trial, right? That we were all very disappointed about. And ALS is not a singular disease. Stop treating it like one. I'll tell you, my personal feeling is that this is the key. And this is where I think you guys can also help, because clinical trials all now have, community collaborations and patient advisory boards. And it's really important that we are rigorous about giving the right drugs to the right people, or we're just not going to figure this out. So, I get asked to get those kinds of talks all the time because of what I do for the DoD. And I mean, literally, and they're often, oh, talk about the pipeline. And I don't like the word pipeline, and I want us to change it. I want us to all stop using the word pipeline. And this is getting back to what I truly think is what's changed over the past decade that wasn't captured in either of those papers, either Dr. Griffin's or that more recent review. A pipeline is a tube that is closed, and it's often an eyesore. It doesn't interact with its environment or its community, and it just goes from one place to the other, and and that's it. And that's the way research used to be done, right? It used to be somebody did something in a lab and they handed it off to a super secret drug company who then had their pipeline, and and the only involvement of people living with the disease or the community was maybe to raise money. And the thing that's changed about ALS is that this is simply not the way we do business anymore. Oh, by the way, this is the other thing that can be like, right, a pipeline for a surfer that's about to crash on him and kill him or knock him over. Again, I don't want to use this word anymore. If you Google the word pipeline, like image search, this is what you get. Not how I want my clinical trials in ALS. This is what we often talk about as a pipeline in ALS and sort of the pathway for a new drug. And so in ALS land, the early sort of risk and epidemiology studies, the CDC, a lot of the basic biology is funded by NIH, NINDS. Hopefully that's going to continue. Fingers crossed. Oh my gosh, it's weird times. The DoD program that I chair the programmatic panel for, we're sort of in this niche right here that's target validation and then, you know, what are called IND enabling studies to do what's necessary to get something to a clinical trial, to get the data needed and the biomarker data to be successful in a clinical trial, and then early phase clinical trials so that when somebody comes to, say, the Healy platform, they've got this stuff in hand. Now, as a community and as a world, we still depend on industry to fund the later stage trials. They're expensive. They cost a lot of money, and they're funded by industry. And the reason I don't like the word pipeline is this. Every single freaking part of this pathway has been majorly impacted by you, right? By people living with the disease, by ALS warriors, advocates, family members. The way that we operate in every aspect of this process has changed because of you. And that is, in my mind, the biggest change and the reason that I'm more optimistic than ever, and the reason I think we have so many amazing therapies that are moving through this process right now. So the DoD program that I chair, you know, the hallmark of it, I know some of you participated, is that every aspect, peer review, programmatic review, designing the mechanisms, figuring out how they're scored and rated involves people living with the disease. The CDMRP calls them consumers. I don't really like that word, but I get it, right? Because it's the community who is consuming what is produced by this program. I was super proud of this. It's not me, right? It's truly not me, but it's this program that I get the privilege of working with, you know, that was one of the first to get awarded for the Morris Principles. Nothing about us without us. The CDMRP ALS Research Program includes people living with ALS and all scientific advisory bodies. Since its inception, it's included people with lived experience of ALS as reviewers of grants. The scores and input of consumer reviewers carry equal weight to that of scientific reviewers. And this is, I think, why this landscape is changing. And what I'm going to ask you in a few minutes is that we now need help. We need you guys to be the voice of making people responsible consumers, and we'll get to that. And so, you know, preclinical research before the clinical trials, like this, our country, the US for decades and decades has led the world in you know, medical research. And it's because of the NIH and this idea of peer review, right? And study sections. And so the first NIH study sections met in 1946. First study section was about syphilis. This was the first time where there wasn't some good old boys network deciding who got the money, okay? That grants were being reviewed by peer scientists. And part of what they did, the reason they were called study sections, was that they also were tasked with reviewing the landscape and deciding what was most promising and what was the things that were maybe going to translate. And you know, this system, it has problems. It's too slow, other things, but it is really what's responsible for us leading the world of medical research. It's maybe in jeopardy right now, and I really hope not. But the issue is, well, one issue with it is that it's slow, right? So it takes a long time to go through and to do this peer review and all these other things. And there's not enough money. And so only a small percentage of proposals can be funded. And reviewers, you know, have to figure out which model might be the best model, which target, which disease pathways. And in a disease like ALS, we just don't know the answer. Like until drugs work, who's to say that one model is better than another? And this is why it's so challenging in our disease. By the way, this is the first study section, 1946. What do you guys think? Diverse? You know, nothing about us without us. Like the only diversity in that room is some of them have dark suits and some of them have light suits. And little history about this too. The guy who chaired this committee, who by the way was from Hopkins, is the person who figured out that penicillin worked for syphilis. So he had some conflict of interest too. But we've come a long way, and we've come a long way because of IMALS and groups, you know, other groups in this space, you know, really making sure that this isn't how we do things. And also, you know, I joined this program here, and we would get 100 applications and could fund 3, and now we're here. And that's, again, because of advocacy that's raised this funding and helped us to design these mechanisms where we can hold researchers, hold industry, hold their feet to the fire, right? If you want funding from us, you have to do these things. You have to have mechanism-specific biomarkers. You have to do these specific things in the design of your trials and your preclinical data that are going to inform and make sure that we get rigorous clinical trials where at least if the trial fails, we know why it failed. And and we can tell if it worked or not. And I've got some links on here. I talk too much. I own it, whatever. But you guys can look this stuff up. Unfortunately, the CDMRP website is down as part of some things going on in our world right now. I will tell you that I have been told unequivocally it is about to come back up, and just stay tuned. And by the end of this week, and there should not be any impact, but fingers crossed that that's true. This is, again, all available on that website. And if you want to be sneaky, like I know all of you are, you can go on the way back machine, the Internet Archive, and find all of these pages too. So the problem is, though, that we still can only fund early phase trials. And by the way, we also this year decided we really are going to also prioritize novel, innovative science, not largely, but in part because we're concerned about NIH, right? Even before this election, back in September, NIH put what their expected funding rates will be for new research programs, R01 grants, and the expected funding rate back then even was 4% or 8%, depending on how much your budget was, basically. And I know a lot of researchers in not just ALS who have decided that's not worth it. Why spend 2 months writing a grant if I got a 4% chance of being funded? And so I really hope that our program is going to be able to continue to fund innovative ideas and clinical trials and the steps in between. But late stage clinical trials still depend on pharma because they're expensive and because we don't have a budget that allows that. There are CDMRPs and other diseases that are much larger than us that do fund clinical trials, like the breast cancer, prostate cancer program. And so here's the rub, right? So pharma we're all on the same team. We want them to succeed. They want to succeed. They want to help us. They invest tons of money in these drugs. They are trying to do rigorous trials. We are trying to help them. But there have been studies looking at this and looking at how trials are talked about if they are sponsored by industry versus not industry. And the most commonly discussed case is this drug donepezil for Alzheimer's, Aricept. And so there have been several reviews of this. So this drug, there were 5 or 6 large late-stage trials. 2 of them were grassroots funded, not by industry. The others were industry funded. The data from all of those trials, whether industry funded or not, looked identical, meaning the actual data sets looked the same. The non-industry funded trial said, this is a negative study. The industry funded trial said, this is potentially interesting, and there's a statistically significant improvement, and it's, maybe and in some people early in disease, it got better a little bit. By the way, 2 of those trials, actually, the scale that they used, yes, there was statistically significant improvement, but it was smaller than the amount of improvement that could be discerned by either a caregiver or a person living with the disease, right? And so was that clinically meaningful? Who knows? But there have been analyses that show, and this is nobody's fault, it's because of the different vested interests, that trials, late-stage trials funded by industry are talked about differently even when the results are the same. Now, people on our programmatic panel for the DoD and otherwise, we always say, we have to punish industry people, companies that overhype, right? That's horrible of them. And the fact of the matter is, we're not going to be successful in that. We're not going to be successful in this massive landscape of getting everybody to never overhype. And they're doing it for a reason, right? They need to have funding and support, and they need to have their programs move forward. And so instead, what we have to do is learn how to be responsible consumers and teach others and educate our community about what's important and what's not. We need to work together on this. This is just an example. So people send me these a lot. This is one from last year. Maybe some of you guys remember it. Breakthrough that can halt the progress of ALS developed by Northeastern scientists. This was a press release. Major breakthrough potentially helps stop the disease in its tracks in as much as half the cases in the US, right? This was a press release. Somebody said this to me. I'm, holy shit, pardon my language, but that sounds cool. So I looked up the paper. This was the paper. There's a link to the paper. You guys can look it up and evaluate for yourselves. And I looked at the paper and I said, wow, I know some of these people. These are really good scientists, like rigorously good scientists. And by the way, this paper is fascinating, amazing science, but it is early chemistry. It is basic science chemistry. And in fact, while they did give this drug to mice, it didn't do anything in the mice. Like the point of this paper was that, look, we can give it to the mice and it distributes and it has a chemistry effect. And we've done some medicinal chemistry work. But this again, remember the figure I was showing you before? So probability of survival. So this is the orange is the placebo and the blue is the drug. And it actually says here somewhere, where is it? Somewhere in this, I'm not seeing it because my brain's not working, but in essence, they very clearly said no effect on progression of the disease, right? And somehow this generated this. And anybody can go look at the paper. And by the way, here's the link for the press release. And I encourage you to do so because I think you'll find you don't need to understand. I don't understand the crazy chemistry in this paper, like not at all. It is really, really deep in the weeds stuff. But I can figure out what was the comparison and what did they do and what was the measurement and what was the model and how did they measure it. And I think anyone without any training can tell this is irresponsible, right? And so that's what we need to do. I think probably everyone on here is familiar with ALS Entangled, another thing that Dr. Bedlex added that a lot of us are involved with. I would encourage you, if you've not been on this website, to take a look at it. We review all sorts of off-label treatments. There's zillions of them. There's a specific way we do it. Some of these have actually resulted in trials because they have been promising. We grade them in a particular way. There's podcasts on here. You can suggest things for future reviews. I wanted to put this up here because of this particular paper, because a lot of the trials out there that are maybe quote unquote, not trials, but therapies that are touted on in this interconnected world we live in, that some might say are snake oil. Although I've recently learned it's not, we don't call them snake oil anymore. We call them essential oils. But anyway, this is a good paper to read. It talks about red flags to look at when you're reading these press releases, when you're reading these websites, when you're reading these announcements about results. And, this is something I really, really want to stress. How do I get my information, right? Where do I turn to? I turn to you, like legit. When I want to know about what's going on in the clinical trial landscape, it is organizations like IMALS and TDI and Everything ALS that have these resources that are my personal source for the information, for the primary information. I also, just a shout out, those of you on Facebook, I love this group. It is moderated by several people with lived experience of the disease, and they do an unbelievable job, better than anyone I know ever could in the research, among the physicians or scientists. It's remarkable. This, again, the website's down, but I have created a resource list that will be right back up very shortly on the DoD website, which has all of the clinical trial search engines, including, ALS Signal and other resources. Check it out once it's back up or find it on the Wayback Machine. I don't know. So this here, I made this tiny URL, and please check it out. This is an unbelievable thing. Maybe actually, you know what? Let me see if I can just share this for a second. I think that's my last slide, and if it's not, it should be. Yeah. So it'll take a while to load, but this is made by a person living with the disease, and it is the most massive thing I've ever seen. And it's not been updated for the past couple of months, but it was being updated weekly. It will be again soon. There's actually one of the foundations that is going to take it on and have the code, but let me just show you here. So it goes by, it's got like all the, like what's available, what's not, what stage, what's the treatment type, what's the biology. The reason I like it myself is that it's got, again, this is sort of hard to maybe project, but every drug under development with links to the references and the websites and the information about them. And let's see here. If I go, yeah, so these are all the different drugs, all the companies, what stage they're at, notes about the science, the biology, the preclinical science, links to the publications. This is made by a person with a disease. It's unbelievable. And so this is you guys doing this, and I'm grateful. And, it's because of this sort of work that we have this really full pipeline, and we're finally starting to see some signals, and I think getting some drugs that are going to really work. I think that was my last slide. It was. So hopefully that was interesting, and I'm happy to answer questions or talk about whatever you guys want for as long as you want.
Thank you. Julie, do you want to see what's in the chat?
Yeah.
and open the floor to anybody who wants to ask a question live.
Yeah, in the chat, Lynn says, crucially, cancer treatments usually target multiple things simultaneously. Multiple myeloma now has 4 compounds. ALS Land continues to test a single compound at a time.
Yeah, Lynn, so this is absolutely true, and it's interesting because they test different compounds, but they have measures to show that those compounds in a person are likely to be beneficial. And what they find when they test these multiple compounds is that often like 1 + 1 + 1 = 9, right? Like if they're targeting different mechanisms in somebody and they know that those different mechanisms are active in a person, that they get more bang for their buck. And certainly, that happened in HIV, other diseases as well. We may be starting to see some of that in ALS, right? Because I've seen some recent studies that suggest, for example, that being on the combination of Riluzole and Edaravone maybe has more benefit than just either one alone or 1 + 1. And so I agree that is something that hopefully in the future we'll be able to do. I still think it needs to be targeted to the right people. And actually, crucially, the one thing that really we should never run a clinical trial where we can't tell if the drug has the biological effect it was supposed to have, because then at the end of that trial, if it doesn't work, we just don't know what the heck happened, right? And we shouldn't accept that. We shouldn't accept trials where there's not a clear measure of did it affect the biology or not, because if we don't have that, we're not learning anything.
Pete.
Yeah, thank you. As we identify more and more genes that are mutated or whatever it is, it seems like suddenly we're just getting all these different roads to investigate. But are some of those, and I didn't know why they were color coded, just similar dots on the same road? In other words, they kind of point to the same mechanism. So there's
Hang on, you mean the color coded on the figure that I showed? Let me pull that up and I'll explain it.
Was that
If I can find that. One
that
second.
curve, I think, that was showing the genes.
Yep.
So
Yeah, good question. I'll explain it to you. Okay, this I think, let me see if I can share my screen again. This is the color coding you mean?
yes.
Okay. So no, so in this figure, but in this figure, those color codes actually mean something else. They mean the point of this figure. So the size of these circles is how common they are, right? So C9orf is the most common, right? SOD is the next most common. These colors are these new fancy schmancy ways of doing genetic sequencing that didn't used to be in existence. And so the point on this figure is that they are these new technologies that are making this thing shoot the moon, that we're finding these genes we didn't find. Now, this figure here, though, is showing that many of these genes are, targeting the same pathways, right? And so when a gene mutation screws something up, like you might hear about something called gain of function or loss of function, right? So the point is this: if you have a mutated protein, either it's not doing something it was supposed to do normally, or it's doing something bad, or it's doing both of those things. And one of the things that we're learning about some of these genes in ALS, the easier ones for us to target with gene therapies are the ones that have what we call toxic gain of function, meaning if it's doing something bad that it's not supposed to be doing, then we can use a genetic strategy to silence it, right? To get rid of it and maybe have a therapy. And so, NLORM and Columbia University has this program right now looking at these super rare mutations and sort of personalized medicine for these super rare mutations. The ones that are most promising are the ones that are these toxic gain of function. The ones where the gene is causing something, losing some normal function, those are more of a challenge, and we're finding that maybe those are going to be harder to target. The thing that is frustrating to me personally is a lot of these genes, again, look at these mechanisms, right? Like every cell in my body does RNA metabolism, okay? Every cell in my body, has to transport things between the nucleus and the cytoplasm, nucleocytoplasmic transport. And how it is that all of these broad genes can cause this very specific disease. And oh, by the way, a lot of these same genes and these same pathways are implicated in other diseases, and not just ALS, and not just brain diseases like, say, dementia or Parkinson's, but prostate cancer or hiccups or migraines. I mean, it's really, really something we don't understand still is what's called selective vulnerability. So how is it that all these diverse things can cause this specific disease? If we could figure that out, then maybe we can get, truly treatments that will work for everyone.
Thanks, Nitesh. Do you have a question?
Yeah, a great talk, Dr. Ostrow. Like a lot to learn. Like I am still learning a lot. So, I have a question. Like, do you think it's a time to change our perspective of a single target uh, in ALS? Because it's been already, known that ALS is a multifactorial disease, and with the mouse, you have shown a lot of preclinical and
Yeah.
mouse in lot of slides. So I have been also doing my studies. So do you think like, that's been lot discovered in the mice, that mice also has a multifactorial pathophysiology. Mice is not a single validated, run by a single validated, target. Say for like there's excitotoxicity, which has been already discovered by some groups. There is axonopathy, there's proteinopathy, there's RNA metabolism. It's everything is in the single mice. So we
Yeah.
need a cocktail of treatment, right?
Right. So 3 different points I want to make about that. So one, sporadic ALS is a human disease. It doesn't happen in any animal, right? In fact, people at one point were looking for, are there specific proteins that are only in people that might be the result of that? And so the animal models that we have, they're genetic manipulations. And so number one, before I get to your question about the multiple treatments, we need better models, right? So SOD1 has a very different pathology under the microscope than other types of ALS. The C9orf72 mice that, the treatments were developed on that then did not work in trials, those treatments in the lab, they fixed the abnormalities that we see under the microscope, but those mice didn't really get ALS. They didn't get weakness the way ALS does. Now, there's several newer models that are in development that look more like ALS. There are some models that are better. So there's a model, Dr. Nath at NIH has made a model of this. You might have heard of this endogenous retrovirus called HERV-K, where they put this envelope protein in the mice, and it really does get a disease that looks like ALS. And, this is there have been some treatment trials looking at these antiretrovirals like for HIV. One of the things they've learned from those trials that it's only about 1/3 of people with ALS that have abnormalities in that pathway. And so those trials aren't going to work unless that drug's probably not going to be useful unless you give it to the right people. And those drugs in HIV, you give a dose and you only have to get like 50%, inhibition in order to get an effect. It looks like in ALS, you need like 99% inhibition. So that, the dosages have to be adjusted. As far as this idea about targeting different pathologies, the real answer to your question is, who am I to say what's going to work until something works, right? But the part that is most frustrating to me is that we have lots of data right now about mechanistic clusters in ALS. I mean, there's several papers that show that if you analyze brain tissue, spinal cord, peripheral cells that are derived from the brain, that they cleanly fall into different clusters, that some people living with ALS have abnormalities of oxidative stress, some have abnormalities of these different pathways. And we know that, right? And we know that our drugs target these different pathways. And so it is a no-brainer to do cohort selection, cohort enrichment in trials of these drugs that we currently have. And the problem, so while I agree with you, the premise that I do believe that eventually we will use a cocktail of medicines to treat somebody with ALS, we're not going to figure out what works and in whom it works if we do the trials that way, right? Because if you do a trial with 4 different drugs, one of them works a little, one of them has a toxic effect that actually makes people worse, one of them speeds up the disease, one of them works a lot, you're not going to be able to sort that out. So I think we have to be rigorous about, again, biological engagement. Can we see the biological effect? And making sure that we are knowing how we're affecting the biology in each person. And then, yes, I do believe that in the future, we will use cocktails of the target multiple mechanisms as a treatment for ALS.
Thank you so much.
In the chat, Michael says the recent trials with clear results, Phoenix Tudka, Adore, were 48 plus weeks and 300 plus participants. The 6-month trials with less than 200 have all had vague results. Oh, 200 more.
Yeah.
All had vague results, and then we're into the post hoc. But the ALS clock makes longer trials difficult. How do we balance this?
Right. Yeah, this has always been the issue, right? And the idea that, some trials, the inclusion criteria where they wanted rapid progressors so that you could detect a change, right? Even though the biology might not, the target might not be the thing in rapid progressors. I'll tell you, it depends on what you mean by balance, okay? If we had A, better measures of biological change, and B, more granular measures of disease progression, we would be able to do shorter trials with less people, right? And personally, as far as the disease progression goes, this is where I think the digital outcome measures and digital biomarkers hold a lot of promise, right? We're able to detect changes very early. We're able to detect changes in people with, at risk with gene mutations, perhaps before they realize they have ALS, with things like, Bluetooth enabled shoe soles or passive data collection from different sorts of wearables. And I think the way to fix this problem is to have more granular measures of disease progression. And then also, so if somebody's going fast or slow, if I'm measuring changes in a particular biology, I should be able to tell if my drug has an effect. Beyond that, there's no magic answer. It's like, how long should a trial be and how many people? There's not going to be one specific answer. One thing that I personally get frustrated by with some trial design is when I see a trial that says, oh, we're going to, because of our post hoc analysis on the last trial, this time we're going to enroll, fast progressors or slow progressors or early ALS, right? These are not necessarily things that are based on biology. What's early ALS? No. Enroll the people who have abnormalities in the biology that you are targeting. ALS is a fatal disease. Why? It's a fatal disease because it eventually affects breathing and swallowing, okay? Rapid progressing is really difficult to define in that context. If your disease starts with your breathing and swallowing muscles, well, guess what? You're more rapid progressing than somebody whose disease maybe that starts in their foot, okay? And doesn't involve above the neck until much later. But more than that, there's not a biological basis for this idea of early disease, late disease. And so I would rather that companies were focusing on matching the biology to the patients and having granular measures of disease progression at whatever stage and whatever rate somebody's progressing.
Thanks. Lynn says, human iPSCs built into mini brains hold much promise.
So agree, and we funded a bunch of those studies with the DoD, the ALS research program, right? And I agree they hold promise because they are derived from people living with the disease. The caveat with that, though, is until we successfully develop drugs based on these models, I don't know that they're the right model, right? That's why, I think we still have to wait and see. We've learned, so iPSCs, so these are induced pluripotent stem cells, right? Originally, we made these out of people's skin cells. So we would take skin cells, fibroblasts, like a skin punch, like a dermatologist does, and we're able in the lab to turn those cells into what are called stem cells, and then turn those into nerve cells in a dish or other types of brain cells in a dish. And they let us do, like a clinical trial in a dish. And we could have those cells from people with different flavors of ALS or normal people and throw drugs at them and see if we made the cells look like each other. And the idea, quote unquote, mini brain is that now there's ways to make this more like the apple pie that I showed you and less like the applesauce. And yes, we can see things in these models that seem to be similar to what we see in people. And I'm optimistic, but until we get drugs that work, we have to wait and see.
I have one last question. So, Dr. Ostrow, do you think a global oxidative stress phenomenon is not a target, but a cellular pathological phenomenon, which is an important factor, like implicated in,
Yeah. Right. So here's the answer. So I don't want to. I didn't. So the analogy that I often use. So I live in a really old house that's really drafty, right? Really, really drafty, and it's a stone house built in the 1930s. And if you didn't know what made a house drafty and you were doing an experiment and you compared my house to the house next door, right? And said, what's different about our houses? The things you're going to find, for example, are we probably burn more oil in our furnace than my neighbor, right? The problem is not that we're burning too much oil. The furnace is working harder because my house is drafty. My furnace may be a great furnace, but it probably breaks down more than my neighbor's furnace. Again, why? Because I'm using it more. It's not that there's something wrong with it. We probably have more ash in our fireplaces than my neighbor, you know? And if you looked at that and said, oh, that's the problem, we have to take the ash out of the fireplace, that's not going to help my house. Everything's connected to everything in our biology, right? Most of those mechanisms, like you just mentioned, oxidative stress, these are general cell mechanisms that are used by everything in our body. It's not surprising that some of these mechanisms are abnormal in people with a chronic disease, and more abnormal or less abnormal in people with different genetic backgrounds. It doesn't mean that it's the cause. It doesn't mean that it is or isn't something to target. For example, my in-laws would come to visit us and they would turn the heat way up and we would burn $80 a day in oil and we'd get pissed off and we'd follow them around and turn it down. And then, you know, one Christmas holiday, we gave my father-in-law these really goofy looking footy pajamas to wear. Okay. And the footy pajamas did not in any way treat the cause of why my house is drafty, which is single pane windows in a stone wall that we can't do anything about. But guess what? He didn't feel cold anymore. Like, that's a good treatment. Okay. That treated the problem. It didn't have anything to do with the genetic mutation, which is the windows in my house. And so you can target things in ALS that aren't necessarily the underlying cause, and in other diseases too. And you know, the point is, when we do a trial and we have a mechanism that we're targeting, we absolutely, every single time, there has to be a measure in those people getting the drug to show whether or not the intended biology was affected, or we're not going to learn the answer to your question. We're not going to learn whether targeting a general mechanism like oxidative stress or, you know, something else like that is worthwhile, and in whom it's worthwhile.
Dr. Ostrow, one last question and then we will let you go.
I'm fine. I talk forever. It's
And
I'm
Elena,
subjecting you guys to the pain, not the other way around. So, you know, yeah.
in the chat, Elena says, John Jumper won the 2024 Nobel Prize in Chemistry for developing an AI model called AlphaFold.
Yeah.
Predicts 240 million protein structures, and it is anticipated that this model could solve many research and health issues. Do you know about John Jumper? And
I do, and I know about, so, you know, in this world we live in right now, machine learning and AI algorithms are being used in different ways in medical research. So one way is to analyze these giant data sets of proteins and RNA and DNA and different sorts of molecular and biological data. Another way is actually to model disease progression, right? To take a video of you on your cell phone and analyze changes in the way that your face moves, or the way that you talk, or the way that, you know, your speech sounds on a phone, and detect things about disease progression. And all of these methods certainly have a lot of promise and are generating a lot of excitement. We've struggled with this at the DoD program in the following way. There's very little, like, you know, we have, there's white papers or guidance documents about how to use animal models in disease research, about how to design clinical trials. There's frighteningly little best practices when it comes to these AI machine learning models. And it's very challenging to evaluate them next to each other and figure out which ones are properly rigorous, which ones are maybe seeing, you know, things that aren't really there, hallucinations, data that isn't actually meaningful. That's an area of our world right now that is also moving forward, but we've added more data scientists to our programmatic panel for just that reason, to see if we can get some better guidelines, better help to researchers to assess these machine learning and AI models and figure out which ones are most likely to bear fruit. But I agree, it's very exciting stuff.
Well, on that note, I think it's appropriate that we end on an image of Dr. Jumper rather than Dr. Denton. But I want to thank you thanks for smiling. I guess I'm showing my age. We just thank you. I think that your devotion to the community, as well as knowledge of research, as promised, was more than evident in this. And we thank you for your generosity of time and attention.
I'm
And thank you
grateful to be
for-
here. Truly, I love you guys. If there's ever anything I can do, I mean it. You know, this is, you know, this is what I've devoted my life to. I don't intend to fail, for what it's worth, but I work for you. And so anything I can ever do, please let me know and reach out.
Thank you, Dr. Ostrow. Take care, everybody.
Cheers.
Thanks very much.